Wave me. I'll try my best to keep it slower. I tend to talk really fast, but I'm going to be, I'm going to try my best. So I'm Marissa. I work in Amsterdam. And this is the hospital where I work. So our lab is located in a hospital, in the Amsterdam University Medical Center. It's a really big hospital. And our lab is called the Laboratory for Genetic Metabolic Diseases. We are about 60 to 100 colleagues. This is our fancy lab with all the fancy machines. So what do we do in our lab? We study inborn errors of metabolism, which are genetic diseases that affect the cell metabolism. This is a very simplified scheme of cell metabolism. So basically, the food that you eat enters your cells, where it gets transformed into the building blocks, into little simple molecules. And those molecules can then be used to make energy, to make all the components that the cell needs. And once they get old and need to be destroyed, that's also the other part of the cell metabolism. We study these diseases at different levels. So we can sequence DNA. We also have functional enzyme tests to actually analyze the function of these proteins. And we also measure metabolites, for example, in urine, but also in blood, in plasma. We work in very close collaboration with physicians and patient groups. Because we are in the hospital, it's easy to contact the physicians. And we also do fundamental research and translational research. So the point of our research is always to give to society, to the patients, to the patient groups. Our path to amino acyl tRNA synthetases, or Nars, started with a question from a physician that had several patients with one of the ARS diseases, and they asked us if we could develop a method to actually functionally prove that those mutations were disease-causing. So that's what we did. From cells from patients, we developed an assay to analyze the amino acyl tRNA synthetase function, including Nars1. So as I said, we do that in cells from patients. So that means that to do our assay, we need fibroblasts. So we need the patients to go through a skin biopsy. So basically, the physician uses a needle and takes a little bit of skin. So it's an invasive test, and it results in a wound and a tiny scar. But that sample for us is really very precious, because those cells, we can grow them. So it's like an endless sample. Also, we can share it between different labs. Different researchers can have access to them, independently of which therapy the patient does. As we grow the cells in the lab, we can control how they grow, so therapy is not going to affect that. And these cells can be used for additional diagnostics or to study therapies, and we can even transform those skin cells into other types of cells. Now, onto our project. Here today I'm going to show you what we will be doing. So we got our funding from the Rory Bell Foundation. We have no data yet, so I thought it would be interesting for you guys also to hear what are we actually going to do. So we are going to use fibroblasts from patients mainly. And we are lucky enough to have access to 8 different cell lines from individuals with Nars1 variants. We also have fibroblasts from 2 fathers. And we're going to use controls like Ilka said, age and gender matched controls. And this grant is divided into 3 aims. In the first one, we want to study the function of Nars1 in health and in disease. With the second aim, we want to specifically study the effect of fever and infection on Nars1. And for the third aim, we are going to look for a biomarker of Nars1 disease in plasma, which is like a part of your blood. So for aim 1, what we are going to do, we are going to do omics. So basically what we're going to do is we're going to look at all the tiny molecules that are inside your cells to see what's going good there and what's not going so good. And then we compare what we see between the different groups. So I'm going to continue the analogy that Ingrid used. Imagine that your body is a kitchen. So the ingredients that are in this kitchen is like your food. The recipes are the genes. And your metabolism is like the cooking process. So with omics, what we do is like we take a snapshot of that kitchen, and with that information, we try to figure out what's being cooking there, the smells, the leftovers, the product itself, is the oven warm? So those kind of things give you an idea what's happening in the cells. And then what we do is we compare the different kitchens. So for example, here you see that the one pizza doesn't have tomatoes. So that's basically what we want to identify what's different and what's not. And by comparing the different groups, we can identify which components are specific to Nars1. And why is this important? Because the more we know, the more we understand the function of Nars1. And if we understand the function of Nars1, it's easier than to identify possible ways to help your cells to restore the normal function. So for the second aim, we want to study the effect of fever. And we do this because we know that for another synthetase, so for the one that does leucine, so for the Lars, we have a group of patients that get really way more sick when they have a fever or an infection. And we have one cell line, well, currently we already have 3 of Nars1 patients that have the same. So when we measure how good the Nars is working, it's not working really bad. But if we grow those cells at a higher temperature, then the activity really goes down. So Nars1 really doesn't like that. And we want to investigate whether that's a more common thing. So basically, in this case, what we're going to do is we're going to grow the same cells at different temperatures. And because we also want to make sure, we want actually to distinguish between fever and infection, we are also going to grow the cells in the presence of like a substance that tells the cells you're being infected. And then we are going to compare all those groups, again with the same omics analysis. And this will show us actually what's the importance of Nars1 during fever, and also if these patients might need extra care in case of disease. And for the third aim, we are going to identify biomarkers in plasma. And actually, what is a biomarker? Biomarkers are basically compounds that differ in amount between groups. Why do we need them? We need them to identify disease. So it can be to accelerate or confirm diagnosis. And also, in case of a clinical trial, you need to have something measurable where you can see if the drug is working or not. Why do you use plasma in this way? Because you can collect it along the time, and then you can follow what's happening in the cells. So basically, this is the type of result we will be looking at. So you have controls. You get that in these cells, you identify 16 different molecules. And then if you compare them with the cells from Nars1 deficient patients, you see here there's one compound that's way higher. So the peak is bigger. That means it's way higher in the Nars deficient cells. So this is our biomarker. So when you have another sample that you think this might be a Nars deficient patient, you can do the same test, and then you can see if compound 6 is high or not. And when you reach the clinical trial level, you can also follow this compound along the time, and then you can see if it's normalizing, if the therapy is working. So basically, in conclusion, with this proposal, we are going to study the importance of Nars1, we're going to study the impact of fever and infection, and we're going to look for biomarkers of Nars1 disease with the purpose of advancing towards a cure for Nars1 disorder. So I would like to really thank the Rory Bell Foundation for the support. We have hired a research technician, Luke, that will start next Wednesday, so really happy. So these are the people working on Nars. Me and Desiree, we are the main scientists doing the work. And then Luke is going to start. Vera is a PhD student also starting. And Chaya is our boss and the PI. So thank you very much.
Does anyone have questions? about what Marissa just covered. I would say a couple of takeaways for me, and we heard a presentation from Marissa and her team 2 years ago at the conference in Canada, and it was the only one that I understood because it was talking about fevers and temperature and the impact on kids with Nars1. And I think it really clicked for me because originally when Grady started having seizures, we noticed they were always when he was homesick from school. So he was, they weren't overly aggressive. He was having like one every month or every so, but something that just kind of like clicked with me in terms of understanding like, oh wow, maybe there is something here. I don't think you had studied it in Nars1 specifically, so I think this will be, great to understand the translation of that. But I do think something pretty common that we've heard from a lot of parents and seen with kids in terms of not only are they really impacted more when they are sick and have fevers, but we're also hearing there's seizure activity sometimes too that's amplified when they are having fevers and just temperature sensitivity as well. I think they're kind of lethargic and things like that. So I think that one really stuck with me for sure. There was something else I was going to say, and now I don't remember what it was too, but thank you.
Yeah.
Any other questions, thoughts on that one?